For more than 20 years, scientists have studied human disease using a single reference genome. But here's the catch. That genome was stitched together from just over 20 people, yet most of it came only from one. And humans, we are not a single drop in the ocean. We are the entire ocean compressed into a single drop. Within each of us lies a universe of variation, too vast to be captured by one blueprint alone. Welcome to the world of bioinformatics.
Medicine isn't a one-size-fits-all solution. For years, scientists relied on a single human reference genome, built mostly from one individual, as a standard for studying disease. But that reference left out most of humanity's genetic diversity. To fix this, the National Human Genome Research Institute at the U.S. National Institute of Health launched the Human Pangenome Reference Consortium, an international collaboration of researchers, technologists, and bioethicists. Their mission is to build a new reference genome that truly represents the global population. The Human Pangenome isn't a single recipe. It's a library of cookbooks capturing the full spectrum of human diversity. With this richer genomic map, we can design treatments that fit individuals more precisely, wherever they come from.
The Human Pangenome is a game changer for global health. The old reference genome, GRCH38, was Eurocentric and incomplete. That bias meant genetic diagnoses were often less accurate for people outside the European ancestry. A more inclusive reference means more accurate science and more equitable medicine. Think Takenouchi Kosaki Syndrome, for example. First identified in Japan, this rare disease is caused by mutation in the CDC42 gene, leading to growth abnormalities, distinctive facial features, brain dysfunction, and immune problems. Recently, a case was discovered in Africa. That finding proved the syndrome exists across population, and it was only detectable thanks to more diverse genomic data. This is the power of the Human Pangenome. It helps us see what we couldn't before.
So, how do we navigate thousands of complex genomic maps? One answer: artificial intelligence. AI can learn intricate patterns, detect genetic variants, predict the impact of mutation, and even suggest potential therapies. It transforms overwhelming complexity into actionable insight. In the case of Takenouchi Kosaki Syndrome, AI plays a role in filtering patient genome data, identifying changes in the CDC42 gene, such as the p.tyrosine 64 to cysteine mutation, predicting its impact on protein function and linking it to clinical symptoms. So with that in mind, doctors can more quickly understand the cause of this rare disease. So, here's the big questions. With AI, could we one day find treatments for nearly every disease, whether caused by gene or mutation? AI gives us precision. The Human Pangenome Project gives us diversity, but progress demands direction.
With the tools ready and the data alive, the next step belongs to us. The real question is, what are we going to do about it?